I was sitting across from a senior bioethics professor at a quiet cafe near campus last week. He was mid-sentence, gesturing grandly about the unfathomable mystery of life's origin. To him, the idea that a chaotic soup of chemicals could just become us was a miracle, a statistical fluke so rare it bordered on the divine. He seemed almost comforted by that complexity, as if the sheer luck of our existence excused the stagnation of his own department, or the fact that their latest research project had been tied up in ethical committee reviews for 18 months. Some things, he said with a soft, satisfied smile, are simply beyond human replication. We should be grateful for the mystery. But then his phone buzzed. It was the afternoon of April 2nd. A headline flashed across the screen. A laboratory had just successfully demonstrated a self-replicating RNA polymerase ribozyme. The miracle had been synthesized. It wasn't a divine spark. It was a successful production run. Watching his face was like watching a slow-motion structural collapse. That mystery he spent his career protecting was suddenly revealed to be high-efficiency chemistry, a physical inevitability. But what was truly striking wasn't the discovery itself, it was his reaction. The excitement you'd expect from a scientist was completely absent. Instead, he immediately shifted into a state of frantic defensiveness. He didn't ask about the chemical substrate or the replication fidelity. Instead, he started muttering about regulatory oversight, diversity impact statements, and whether the breakthrough had bypassed the proper institutional frameworks. While the fundamental code of existence had just been cracked, he was busy trying to build a bureaucratic cage for a bird that had already flown. That instantaneous shift from romanticizing life's mystery to desperately trying to litigate its progress, felt like a perfect microcosm of a deeper rot. It suggested that while one culture treats life as a fragile, accidental poem to be protected by committees, another sees it as a logical, inevitable piece of engineering to be mastered. It made me realize that this scientific breakthrough is more than just a headline. It's a civilizational verdict. In a universe governed by entropy, the side that treats reality as an engineering problem will always outpace the side that treats it as a philosophical debate. The question isn't just how life began, it's who has the discipline to ensure it continues.
Right, and that civilizational verdict, it's already being executed. I mean, the convergence we're seeing right now between biotechnology and artificial intelligence, it has completely irreversibly shifted biology.
Yeah.
It's no longer this field of observational discovery where we just look at things through microscopes. It's a field of brutal, output-oriented design.
Output-oriented.
And it's not just a scientific shift. We are looking at a literal global race for biological supremacy, and it's being relentlessly fought between competing systems of governance. Right. The stakes here are about dictating which global power is going to engineer the future physical realities of, our health, our agriculture, our national security.
So it's basically a winner-take-all scenario.
Because the alternative is being the power that's left just trying to regulate a paradigm that has already made your entire institutional framework completely obsolete.
And the physical reality of this engineering mindset, it isn't something you find in academic journals. It's happening on the front lines of global biomanufacturing hubs.
Yeah, the raw operational reality is completely different from the theory.
Totally. To really understand the human friction driving these geopolitical gears, you have to strip away the abstractions. Like, consider Dr. Chen. She's a lead researcher operating inside a really high-intensity synthetic biology cluster over in Shenzhen.
Okay. Yeah.
You have to picture the environment she's in during a final 72-hour, what they call a conversion sprint, for a new modular RNA-based biosecurity sensor.
72 hours straight.
Yeah. And the space is it's dominated by this relentless low-frequency hum of high-throughput sequencers, just these machines running continuously. They're reading billions of base pairs of synthetic genetic code without any human intervention.
Just massive automation.
Exactly. And the air in that facility, it carries this distinct, really sharp smell. It's like stale instant coffee mixed with the ozone from all the heavy machinery.
And that kind of environment exacts a massive physical toll on the people inside it.
Right. Like, Chen's hands literally shake slightly. It's this physical manifestation of just sustained caffeine intake and the totally crushing weight of her metrics.
Her KPIs.
Yes. She operates under a key performance indicator that measures lab-to-factory latency.
Wow. Lab to factory.
Right. And she isn't measuring success in years or even months. She is measuring the time it takes to move a genetic sequence from a computer model to a mass-produced physical sensor in days.
Which is, I mean, it's crucial to understand what she is actually building during that sprint,
Right.
because an RNA-based biosecurity sensor, it's not a mechanical device made of plastic and wires.
It's a biological.
Exactly, it's an engineered biological molecule. The sequence is specifically designed so that when it encounters the genetic signature of a target pathogen out in the environment, it physically binds to it.
So it's looking for a specific threat.
Yeah, and that binding event, it triggers a structural change in the RNA itself, which then produces a measurable signal. Like a fluorescent glow or something. And to engineer that, her team has to digitally design the RNA, chemically synthesize it, and then completely optimize it so it can actually survive outside of a perfectly controlled lab environment.
Which is incredibly hard.
It requires this unbroken, highly efficient pipeline from the bioinformatics software directly down to the chemical synthesis steps.
Right. And during the middle of this intense sprint, Chen actually took a video call with an old colleague of hers who's based in Boston. And the contrast between their two realities, it really is the core of this whole civilizational verdict.
So,
Well, the Boston colleague spent 40 minutes meticulously discussing the ethical framework of the sensor's potential deployment.
40 minutes.
Yeah. And Chen listened to this whole presentation, but simultaneously on a secondary screen, she was actively approving 3 different patent filings.
Oh, man. Multitasking at the highest level.
Exactly. For Chen, the concept of humanity in her work isn't located in some philosophical debate. It's located in the sheer exhaustion of ensuring her nation does not fall behind. Like, her core motivation is driven by shame, but not over lack of ethical discourse.
Right, she doesn't care about the ethics committee.
No. Her shame is rooted in 3 hours she recently lost to a minor calibration error on the sequencing floor.
Wow.
In her operational reality, if you are not building the biological machine, you are simply the fuel it consumes.
Yeah, and the physical and psychological reality experienced by Dr. Chen, it is entirely backed up by the macroeconomic data that tracks this commercialization race. Yeah, so historically, the United States maintained totally undisputed leadership in the field of biotechnology. I mean, if you look back at the 1940s, the U.S. Department of Agriculture, working with the private sector, they discovered the deep tank fermentation methods to mass produce penicillin.
Oh.
They didn't just find the mold, they figured out how to engineer the actual chemical environment. You know, controlling the oxygen, the pH levels, the nutrient feeds to scale it industrially. And that fundamentally altered modern medicine.
And then insulin in the 70s, right?
Exactly. In the 1970s, American biochemists achieved the first successful cutting and joining of DNA fragments. They took the human gene for insulin, inserted it into a plasmid, put that plasmid into an E. coli bacterium, and literally turned the bacteria into a microscopic factory.
Genentech.
That led directly to Genentech and the mass production of synthetic insulin. And by the 1990s, the U.S. spearheaded the Human Genome Project, successfully mapping all of human DNA.
But that historical dominance, it's really fractured now. The current landscape in the West is basically defined by an acute inability to commercialize these innovations efficiently.
It's a huge problem.
The physical infrastructure, like the massive bioreactors and the downstream processing facilities, it's all aging. And long-term capital investment in high-risk physical biology is really scarce right now because the market signals are just confusing and so heavily regulated.
Right. And you have to contrast this with China, which recognized biotechnology as a foundational strategic priority over 2 decades ago.
Decades.
Yeah. And the metrics demonstrating this shift are they're staggering. Between 2016 and 2021, the market value of Chinese biotechnology firms experienced a 100-fold increase.
Wait, 100-fold?
100-fold, reaching $300 billion.
That is insane.
And by 2019, their global share of pharmaceutical output had hit nearly 25%. Furthermore, the share of clinical trials launched by biopharmaceutical companies headquartered in China, it skyrocketed from a mere 3% in 2013 to 28% in 2023. These aren't incremental gains. They represent a systemic, aggressive restructuring of the global bioeconomy, moving rapidly from basic research into industrial-scale production.
Yes, we are basically looking at a scenario where the United States possesses immense theoretical knowledge and raw scientific capability, but the system is trapped within a regulatory thicket.
Yes.
So is the U.S. regulatory environment essentially an innovation traffic jam? Like, if you view the biological products being engineered in Western laboratories as state-of-the-art, high-performance vehicles, the roads they must travel on are unpaved, tangled, and blocked by endless toll booths.
Right. Exactly.
Meanwhile, competing nations are simply building high-speed rail networks, specifically designed to transport these ideas directly from the laboratory bench to the commercial market without any friction.
That traffic jam analogy, it precisely captures the structural bottleneck, particularly when you look at the supply chain economics. Well, United States biotechnology firms operate in a highly complex regulatory maze. Agencies face persistent understaffing despite a continuously growing backlog of applications.
Right.
Innovators end up waiting years, sometimes a decade or more, just for approvals on novel biological products. And this creates an impossible business environment because in the biotechnology sector, a lot of products are business to business.
B2B?
Yeah, B2B. They are engineered enzymes or specialized proteins or synthetic genetic circuits that serve as ingredients for other downstream manufacturing processes. And the buyers in these supply chains are extremely sensitive to delivery delays.
Right, because if the engineered enzyme that's required to manufacture, say, a new class of industrial adhesive is delayed by a regulatory review for 3 years, the entire adhesive manufacturing line is totally paralyzed.
You lose the whole line.
Yeah.
Furthermore, investors in the current economic landscape, they've become highly risk-averse. High interest rates, combined with the failure of previous biotech ventures to actually navigate these regulatory mazes and produce rapid returns, it's dried up the capital.
So no one wants to invest.
Right. And when foreign state-backed firms enter this equation, they fundamentally alter the market dynamics. They bring fully commercialized products and services to the global market at prices that effectively have no floor.
Because of the subsidies.
Exactly. Because they are heavily subsidized by state initiatives, they can rapidly expand their manufacturing capacity, completely circumventing the regulatory delays that paralyze U.S. firms.
Wow.
Consequently, American investors are terrified of the regulatory queue. They know that by the time a U.S. product clears the legal hurdles, a foreign competitor will have already saturated the global market with a cheaper, immediately available alternative.
Yeah, the high-speed rail has already delivered the cargo, while the high-performance vehicle is still just idling at the toll booth.
Good.
And, the speed of commercialization happening in places like Shenzhen is directly connected to a fundamental ideological shift.
Yes.
This shift, it basically requires systematically stripping biology of its divine mystery and treating it purely as executable code. Like, there is this perspective from Professor Aris. He's a European-born molecular biologist.
Right.
He moved his entire laboratory to an Eastern tech hub about 3 years ago, and his experience really illustrates the profound, unsettling pragmatism of this new era. He described the exact moment he realized the divine spark of life was actually just a cold, thermodynamic certainty.
And this brings us directly back to the self-replicating RNA polymerase ribozyme that we mentioned earlier.
Exactly. It was an intellectual realization that felt like a literal physical blow for him. He recounted this specific evening spent entirely alone in his new facility, and he was just staring at a high-fidelity simulation of it.
Yeah.
And as the hours passed, he watched the digital representation of the molecule fold and bind. He realized the mechanism on his screen looked less like a miracle of creation and way more like a nanoscale assembly line.
An assembly line.
Yeah, and he admitted to experiencing a deep intellectual terror when he observed how his new Eastern students approached the discovery. Like, they didn't see a profound mystery requiring philosophical worship. They saw a highly efficient blueprint for natural nanotechnology.
Right. And to really understand why this terrified him so much, you have to understand the mechanics of what a self-replicating RNA polymerase ribozyme actually is. Sure. So in standard biology, DNA is the hard drive. It stores the information. Proteins are the mechanical engines that actually do the work of the cell. And RNA is usually just the temporary messenger. It carries the instructions from the DNA to the protein-making factories.
Okay, standard biology.
But a ribozyme is different. It's an RNA molecule that also acts as an enzyme, an engine. It can catalyze chemical reactions on its own.
Wow.
Yeah. So a self-replicating RNA polymerase ribozyme is a strand of RNA that can literally read its own sequence and assemble free-floating chemical building blocks to build an exact copy of itself.
It builds itself.
Exactly. It is the ultimate closed loop. It acts as both the blueprint and the machine reading the blueprint.
That is wild. And, Aris felt this lingering sentimental attachment to the Western paradigm of science as wonder. But as he walked through his heavily funded state-of-the-art facility, the reality of his environment just completely dismantled his sentimentality.
How did you put it?
He realized that wonder is a luxury reserved for those who are stagnant. He felt like a priest standing in the center of his temple, just watching a highly organized team of engineers systematically dismantle the sacred architecture to build a functional power plant.
That is a brilliant metaphor.
Right. And the deepest irony was his realization that he was the one handing them the wrenches simply because the power plant actually works and his temple didn't generate electricity.
Wow. And the mechanics of this shift, from a temple of wonder to a power plant of engineering, they are rooted in historical precedent. Biology is currently undergoing the exact same fundamental transition that chemistry underwent back in the 1880s.
In the 1880s?
Yeah. Before the late 19th century, chemistry was largely observational. Scientists just documented the properties of natural materials. But then the advent of chemical engineering unlocked the ability to actually manipulate molecules at scale.
Oh, like making plastics and stuff.
Exactly, leading to the synthesis of rubber, plastic, synthetic fibers. It entirely transformed global society and industry. And physics underwent a really similar transition in the 1940s, right? Moving from academic theory regarding atomic structure and quantum mechanics to the applied engineering of the atomic bomb, semiconductors, and modern computing.
And today, biology is making that exact leap, driven by the convergence with artificial intelligence. Because for decades, the sheer complexity of biology limited our ability to engineer it. Like, a single human genome contains roughly 20,000 individual protein-coding genes.
Yeah.
And these genes produce proteins that perform multiple highly specific functions across hundreds of different cell types.
And the really critical part is that the function of any protein is entirely dictated by its three-dimensional shape.
Right, the folding.
Exactly. A protein begins as a linear string of amino acids, driven by complex atomic forces. We're talking hydrophobic interactions, electrostatic repulsions, hydrogen bonding. That string folds into a highly intricate 3D structure.
And understanding exactly what shape a specific sequence of amino acids will fold into is crucial, right? Because it determines how that protein will behave, what it will bind to, what biological mechanisms it's going to trigger or inhibit.
Yes. This is known as the protein folding problem, and it was a recognized grand challenge in biology for over 50 years.
Because the scale of the math involved is just staggering. By human calculation alone, attempting to enumerate all the possible shapes of a single protein is functionally impossible.
There are an estimated 10 to the 300th power possible configurations for a standard protein chain.
10 to the 300th.
Yeah. To put that in perspective, calculating every single possible fold would take longer than the age of the known universe.
Which is why it's so wild that in 2021, the artificial intelligence system AlphaFold actually solved this problem.
Yes. AlphaFold changed everything.
And it didn't calculate every possible shape using brute force physics, did it?
Instead, it used deep learning algorithms to identify profound evolutionary patterns across vast databases of known protein sequences and structure. By recognizing how certain sequences co-evolved, the AI essentially learned the underlying syntax of biological folding. AlphaFold accurately predicted protein structures within the width of an atom.
The width of an atom.
And this is not just a computational trick, it is the digitization of biological intent. We no longer have to guess how a biological machine will assemble itself. We can accurately predict it, and therefore we can design it.
Furthermore, this transition really aligns with the formal concept of the replicator optimization mechanism, or ROM.
Ah, ROM, yes.
In the original literature, ROM is described with some seriously dense terminology. They call it a mathematical bookkeeping apparatus for scale-relative persistence condition dynamics. Right. But we have to strip away the jargon to understand what this actually means. ROM is not a grand philosophical theory of everything.
No, it's not. It is simply the cold mathematical reality of how things persist in reality.
Exactly.
Persistence condition dynamics is just a very formal way of stating that things that are good at surviving and making copies of themselves will eventually dominate a system.
So, like, if you have a pool of chemical resources and one specific RNA ribozyme can copy itself 10% faster or with fewer errors than another, that faster ribozyme will just consume all the resources.
Soon, 99% of the pool is made up of that efficient sequence. ROM formally proves that once a replicating system exists, whether it's a string of RNA, a human institution, or a digital algorithm, the processes of selection and transmission are not divine mysteries.
They're math.
They are mathematical inevitabilities. The system automatically optimizes for its own persistence, completely indifferent to human philosophical interpretations of its existence.
Interesting. By viewing life simply as an algorithm or a piece of natural nanotechnology governed by the replicator optimization mechanism, we are completely removing the The friction of reverence.
The friction of...
Yeah. If a biological cell is no longer a miracle to be pondered, but simply a programmable computer that executes thermodynamic commands based on a genetic sequence, the only limit to what we can build is our compute power. We are taking the software of existence and running it through an industrial compiler.
And if life is an engineerable machine and the Temple of Wonder has been fully dismantled, the next logical question is, what are these massive biological factories actually building?
Right. And this transitions directly into the concept of biosecurity and the raw mechanics of national survival. Consider the reality of Zhao. He's a 24-year-old bioprocess engineer.
Okay.
He works on the entropy resistance project.
Yeah.
This is a massive state-directed effort to harden domestic agriculture using advanced synthetic biology.
Synthetic agriculture.
Yeah. And his physical reality is stark. His skin is chronically sallow from a severe lack of sunlight due to extended, grueling laboratory shifts.
Just brutal hours.
His social life is functionally non-existent. He harbors this deep, quiet fear regarding the institutional decay he observes in the West. He views the Western Hemisphere as a system that has forgotten the fundamental mechanics of building, entirely distracted by its own cultural reflection and bureaucratic paralysis.
And his vulnerability is entirely tethered to a crushing sense of responsibility. Zhao views his daily work on RNA-based crop resilience not as a career path, but as the active construction of a biosecurity barrier protecting his nation against a collapsing global order.
He described a specific moment of near breakdown when a critical batch of synthetic agricultural catalysts failed in the bioreactor.
What were the catalysts for?
They were engineered enzymes designed to optimize carbon fixation in crops, essentially reprogramming plants to grow faster with significantly less water.
Huge.
Yeah, and when the batch failed, he didn't fear for his job security or his academic standing. He felt a profound personal failure, believing he had actively weakened the civilizational shield.
The coarseness of his daily life is striking. There are no grand speeches in his facility. There are no ideological debates. There is only the gritty, repetitive reality of an engineer monitoring pH levels and nutrient feeds. An engineer who believes that in a universe strictly governed by cold chemistry, the only actual sin is inefficiency.
No.
The rise of his nation is not viewed as a political choice or a policy platform. It is viewed as a mandatory survival reflex against civilizational entropy.
And Zhao's fears regarding the civilizational shield are highly rational when you analyze the securitization of biotechnology. Like, we have rapidly moved from viewing biotechnology as a developmental tool for medical and agricultural improvement to viewing it as a critical, hard power strategic asset.
This shift introduces the dual-use security dilemma. The dual-use nature of biotechnology means that the exact same equipment, knowledge, and processes used to synthesize life-saving therapeutics can be slightly modified to engineer devastating pathogens or toxins.
Same exact equipment.
The bioreactor that brews synthetic insulin can, with a different engineered yeast strain, produce a lethal neurotoxin.
Terrifying.
And when nations implement measures intended to protect themselves, such as restricting international data sharing, implementing strict export controls on advanced DNA sequencers, or blocking foreign investments in domestic biotech firms, it creates profound uncertainty.
Yeah, because it masks their true capabilities and intentions.
Exactly. This lack of transparency triggers deep insecurity in competing states, leading to highly competitive techno-nationalist arms races.
And the legislative landscape reflects this exact escalation. The United States enacted the Biosecure Act, explicitly designed to limit federal agencies from procuring technology or contracting with foreign biotechnology companies deemed threats to national security. The focus is heavily on entities associated with foreign military apparatuses. Additionally, the Biosecurity Modernization and Innovation Act was introduced to broaden the national biosecurity framework to harden the domestic supply chain against foreign adversaries.
Concurrently, an intense intellectual property war is underway. The data from 2019 marks a critical inflection point.
What happened in 2019?
That year, China surpassed the United States in international patent applications, capturing 22% of the global total.
Wow.
And a significant portion of these applications heavily focused on agricultural CRISPR patents. CRISPR, the technology that allows for precise editing of DNA sequences, is being aggressively applied to secure the food supply.
And they're playing defense too.
Oh, heavily. Simultaneously, China utilizes its domestic regulatory system as a strategic weapon. They mandate larger than normal sample sizes and extended testing periods for American-developed agricultural seeds.
Just to stall them.
Right. This deliberately delays market approval, while domestic scientists use that time to reverse engineer the American seed varieties, effectively copying the genetic modifications in a fraction of the standard developmental timeline.
Is this dynamic essentially identical to the geopolitical war we witnessed over Huawei and the rollout of global 5G networks, but infinitely more dangerous because the hardware in this scenario consists of living, self-replicating organisms and the foundational global food supply?
What's fascinating here is that treating biotechnology as a heavily guarded strategic asset fundamentally fragments global research ecosystems. When a nation weaponizes the biological supply chain, the implications are profound. Imagine a scenario where a breakthrough, highly effective cancer treatment is developed, but a nation hoards the required precursor chemicals or the specific biomanufacturing capacity during a geopolitical conflict. By threatening to sever access to life-saving therapeutics or resilient agricultural seeds, you force adversaries into a corner. They have no choice but to build entirely sovereign, completely isolated bioeconomies.
So no more global collaboration.
Actually, the global network of scientific collaboration collapses, replaced by fortified silos of techno-nationalist biological production.
And this biological arms race inevitably extends beyond agricultural shields and economic blockades to the literal battlefield, pushing the absolute limits of future engineering.
Yes.
The economic scale alone dictates military integration. By the end of this decade, biomanufacturing is projected to be utilized extensively across more than a third of traditional manufacturing industries.
A third?
Yeah, we're looking at an impact on a global value chain of nearly $30 trillion. When materials like advanced plastics, specialized military-grade textiles, and even structural biopolymers can be grown in industrial vats rather than extracted from the earth and processed through vulnerable global supply chains, the logistical nature of warfare fundamentally changes.
Consider the application for forward operating platoons. The military is actively developing technology that will allow units to deploy with backpack-sized biomanufacturing systems.
Wait, stop there, because we need to explain how a backpack synthesizes complex biological materials in a war zone.
Right, so it utilizes cell-free biomanufacturing.
Cell-free?
Yeah. Instead of requiring massive stainless steel vats filled with living, breathing bacterial cells that need careful temperature control and oxygenation, scientists extract only the cellular machinery.
So they take out the important parts.
They isolate the enzymes, the ribosomes, and the energy molecules and freeze-dry them into a stable powder.
Wow.
Inside that backpack system, a medic simply adds water, basic carbon or nitrogen sources, and a specific synthetic DNA template. The extracted machinery wakes up and executes the code, churning out the target protein.
Which means instead of relying on highly vulnerable convoy routes to deliver medical supplies, these forward units will synthesize precisely calibrated food, specialized munitions, and shelf-stable synthetic blood directly on the front line. The requirement to refrigerate and transport specific blood types across thousands of miles of hostile territory, constantly defending those supply lines, is entirely eliminated by on-demand biological synthesis.
But this technological leap carries a significantly darker side, explicitly tied to military-civil fusion strategies.
MCF.
Yes, MCF. Military-civil fusion systematically eliminates the traditional, ethical, and institutional barriers separating civilian academic research from the military and defense industrial sectors. This erosion of boundaries rapidly accelerates the potential exploitation of human biological enhancements. The precedent already exists. In 2018, a biophysicist successfully created the world's first genetically modified babies, utilizing CRISPR technology to heavily edit their genomes to resist specific viral infections, specifically HIV.
And this wasn't just a rogue medical experiment. It functions as a clear precursor. It demonstrates the technical feasibility of altering the human germline.
Exactly. Extrapolate that capability into an MCF environment. We're looking at the active engineering of future troops for optimized cognitive intelligence, drastically reduced biological need for sleep, or extreme resilience to radiation and harsh environmental conditions.
Yeah, and to prevent falling irrevocably behind in this operational environment, nations must solve specific, highly complex scientific hurdles known as grand challenges.
Unlocking these leap-ahead capabilities requires addressing the raw, fundamental mechanics of biology. The first grand challenge is achieving a massive deepening of our understanding regarding RNA biology's structure and function.
Right.
As we discussed, while DNA is the static hard drive, RNA is the active functional machinery of the cell. Comprehending its vast array of functional forms is absolutely required to unlock advanced biological engineering.
Second, we must fully characterize all metabolites. Metabolites are the incredibly small chemical components, the sugars, amino acids, and lipids operating within living cells.
And current analytical techniques make it prohibitively difficult to map this entire chemical landscape simultaneously. It's like trying to map every single bolt, screw, and wire in a massive factory while the factory is running. Identifying all of these small molecules will reveal exactly which novel materials can be produced by systematically reprogramming different organisms.
Third, researchers must understand and harness quantum effects within biological systems. For instance, the electron transfer process occurring during photosynthesis in plants operates on quantum mechanical principles. Plants achieve near-perfect efficiency in transferring energy from a photon of light to a chemical reaction center by utilizing quantum coherence.
And mastering that specific mechanism could lead to entirely novel, hyper-efficient energy production methods. It could also lead to new ways to navigate using quantum biological sensors, completely removing the reliance on fragile satellite constellations like GPS, which are highly vulnerable in a geopolitical conflict.
Finally, the ultimate grand challenge. Science must successfully build minimally synthetic cells across various domains of life, including microbes, plants, and animal cells.
By constructing a synthetic cell entirely from scratch, utilizing only synthesized components, researchers guarantee they understand every single basic building block of life. This transitions the field entirely from reverse engineering existing messy natural organisms to forward engineering precise, novel biological architectures designed for specific outputs.
We're looking at a future where the entity that controls the biological data fundamentally controls the physical world. The defining resource of the 21st century is no longer crude oil, and it is no longer silicon. The defining resource is composed of massive, hyper-accurate genetic sequence databases and the raw computational infrastructure required to process and compile them into physical reality.
The self-replicating RNA ribozyme was not simply a scientific milestone to be celebrated in journals. It was the ultimate proof of concept, demonstrating that mathematically driven order and high-fidelity replication are the only forces capable of resisting universal decay. The global landscape is currently fracturing into two distinct models of existence. One side views the power of biotechnology through a highly cautious lens of procedural justice, heavily focused on bureaucratic risk mitigation. The opposing side views biotechnology through the brutal, results-oriented logic of nature itself, where efficiency, scale, and output are the only recognized metrics of success. The side that internalizes the operational logic of the replicator optimization mechanism, understanding that persistence is defined solely by the ability to rapidly adapt and manufacture at scale, is constructing an unassailable strategic advantage.
The pursuit of life's origin is the highest stakes applied science in human history. Life is an algorithm, and compute power is sovereignty. Energy is sovereignty. A civilization that cannot effectively manage its own laboratories has forfeited its right to ponder life's mysteries. In a universe governed by entropy, the side that treats reality as an engineering problem will always outpace the side that treats it as a philosophical debate.